So I found it. So Didem is a senior lecturer in the chemistry department at Boston University. She teaches undergraduate chemistry, biochemistry courses, particularly for pre-health and allied health science majors. Her current research focuses on developing and incorporating authentic tools in teaching biochemistry, but in a large classroom setting. And I think we will get a glimpse of that in her presentation today. So Didem, take it away. The floor is yours.
Thank you, Shuchi. So hi, everyone. This is really awesome to be able to have these conversations. So as you're going to see, I put together some things as more as reminders for me and some things for you to look at, but I am thinking of it more as me telling you a few of the crazy ideas I've been trying to do in class. So the title was AI versus AI because as many of you feel, we're chasing the technology and the way the students use it and what we can do with it, trying to teach them to use it as a tool rather than to just get things done. I want to just give a very brief background of the setting for all of this work. From a course perspective, as Shuchi said, I have a lot of pre-meds, pre-dentals, and some all pre-healths in my juniors and seniors one semester biochem course, and what you're going to see is from them. And also I've been very lucky to be a part of both Molecular CaseNet and Biomol Viz over the years. So a lot of what you're going to see is me trying to utilize a lot of molecular structures in my teaching and assessment. Now, one of the things over time that I struggled as everybody goes towards more and more multiple choice questions in these big classrooms, I still wanted to keep some open-ended questions. And I'm also crazy to have 200 something students and have them write answers or do. So there were projects, then I dropped the projects, but I still wanted to keep some creativity. And yeah.
I just wanted to make sure that you weren't sharing, like you haven't yet shared slides.
No, I have not shared slides.
Good.
Because the first slide you'll see is just one thing, and I wanted to give a context to that. So as many of you teach pre-meds, they want to know exactly what's going to get them those points. So I've been trying to formulate a way to tell them what a good free response answer for biochemistry is. Not just the right answer, but what else can be. So over the years, I tried to come up with some acronym that could help them. And that was, let me now start actually sharing. Can you see my slides right now? Mm-hmm. Okay, excellent. So I try to tell them there are three things, like five C's that you have to think about. One, that it needs to be correct, but I broadened it by saying it also needs to be concrete. I hate them when I say, I think this is the answer. It's either the answer or not the answer. Complete is also that there needs to be coherent transitions between those ideas, not bulleted ideas. Clear and concise requires that they use a specific language that they're building over time. It needs to be specific to the context, and I need to understand what concepts their answer is based on. So to test those kinds of ideas, okay, that's all right. I wanted to give them these kinds of questions. And last year, I shared something about these types of questions and how I used AI, was I would usually give them something like a small background, something to look at. And typically what I would say, based on this kind of information, tell me X, Y, Z. And that was more of a structure function type of questions. And they are used to seeing this kind of question across all the different tests they have over the semester.
Hey, Didem, sorry to interrupt again. Your slide that you're showing, it's not in the full screen view, and two, it is showing a show subtitle something on your.
Okay, let's see.
I think your secondary display got shared because it was like the presenter view.
Okay, let me see if I can. We thought we tested it. Okay, let's try this. And then if I go to share, like slide mode.
Yeah, if you click that.
Does this work now or not?
It's still in the.
Okay, so what I'm going to do is I'll come out of presenter mode, maybe. I'll just run the slides because I don't know why it's doing what it's doing. Let's see if I make this. Sorry about that.
No worries. This is a friendly bunch. We understand. I think every single one of us has experienced this at one point in time in a class, in a meeting, presentation. So we all understand.
Now it's when it's trying to share, it's doing some weird things. Let's see. Okay. Okay, can you now see? I know it's not slide by slide, but you can see the screen, right? At
We
least.
can see the screen slide at a time, and I think we're good.
Yes, I think I'll just stay here. I just wanted you to see the structure. There's a structure and then something that they normally write. And as you might imagine, they wrote everything under the sun. Grading was very, very difficult. And I was trying to get them practice this thing. And this is where the AI comes. Every time I give these kinds of exercises to them, I started realizing they're just asking AI to write the answers. And initially it was not too bad because AI couldn't really understand the images. So I had given them an activity last year to describe the image to the AI so that they can get the answer. And I had some interesting feedback from students where they realized some of the problems with AI. So the major problem with AI's response was that it lacked specificity. So they realized if they don't feed the AI with specifics, we get correct but not very specific answers. So this year, now that AI can also interpret images, and I have to say it does not a bad job, I started trying to build in some things in the class where I was using AI to make questions and the possible answers. So I was sourcing from students' old answers, but also what I found on internet. So something like this. Maybe some of you guys use Top Hat. I do that. That's a clicker system. The reason I like this platform is it has this kind of a setting where you could click the target for an answer. So it works really well with molecular visualization type of questions. So typically I would say, based on the information given, write me something about the zinc finger domain. Instead, I actually wrote an answer and I told them, find the mistakes. And I'm going to show you how I have developed this into a bigger questions for the exams, but also my hope is that it's not only about finding mistakes, but also some biochemical misconceptions, wrong word usage, all of these other 5 C's that follow. I wanted to start with this because I did this in class, and this is what I got. And actually, for many of us who look at these molecular structures, and this is after I taught all the protein sections, like this is after we're done with proteins. The fact that they think there is there are some of these problems here that are so obviously wrong that they didn't pick up, or that zinc finger doesn't have a magnesium. A lot of people didn't get that and all that stuff. So I realized definitely this was a nice way to see what they don't see. So I had given in class, I wanted to write a similar exam question. So I went online and I was asking ChatGPT, find me a small protein. I wanted to have this kind of a 1 helix, all these some sort of criteria. And a good candidate came up as ubiquitin, which was good because I'm going to finish that with that also. So I did first a little search, Google search, to see what kind of things there are on the internet. And I wanted to highlight here, my goal is to see if students are prepared to consume the information they see, and also if they can sort of find out what's not a useful or incorrect information. So I saw there was all these kinds of things about ubiquitin. So I took some of it, like this last one. I took the sequence and then I made my own images. to highlight some of the things that I wanted to really ask for. So this question was more about primary, secondary, tertiary structures and those structural features. So I turned it into something like this, where I gave them biology, more of the biology, and I tell them all biological information is correct. Don't worry about those. And then 3 or 4 sentences with some mistakes in it. So I'll show you how I did this for all biomolecules. This was more involved because proteins are sort of my favorite, so I took a lot of time making these figures, but I wanted to make sure that there was something about their primary structure as well as some other details here. And honestly, I think this is something I wanted to share with you. Switching from a free response to this kind of still select what's wrong kind of thing, I thought everybody will just get A's. This will be so easy for them. I cannot tell you how badly they did on these. But one thing that was very powerful after I ran this both in class and the first exam, I got a huge buy-in that they should not be asking AI all the time to give their long answers, or they should be very critical about those. And yes, we can all write these kinds of questions. I think we all do. I think for me, where AI helped was I had some mistakes and some kind of criteria I wanted to build in. I didn't want it to be too wordy. I wanted it to represent some of the common mistakes students had. And AI was able to save me a lot of time to get everything I wanted in 4 or 5 sentences. As you see, I love talking, so I love writing. So it's usually a big, long chunk for me, but it really helped me to tidy them. And I want to make sure I leave you guys with some time for questions. So I'm just going to show a few other things. So this semester for a different project, I had 4 quizzes instead of exams, one after each biomolecule. So I repeated the structure, but I wanted to show you the different type of images I tried using. So my goal was it should always have an image, and hopefully for next year I want to add graphs in it too, because that's a big problem too. It should have something they need to learn, and then again find the problem. So this was carbohydrates, where they had a lot of issues with stereochemistry, and I think they engaged much more favorably with these kinds of things, but still had to figure out all their stereochemistry. The third one was more about hydrophobicity, lipids, and so on. I love this. These were all my old questions from open responses, but I wanted to hear, not only do they find some mistakes, but in this blue part, they have to select one of the 2 options so that there is a conceptual idea here. So this was more about if the structure did not follow XYZ, the function wouldn't happen. This was more of understanding their enzyme and catalysis, difference in binding versus catalysis. That was what I was trying to teach here. And then this one, the end of the semester, was more towards what I want to go further as I try these types of questions. This was a transcription factor. Typically, again, I ask what are some of the molecular characteristics that need to be there for this kind of function. This time, I sort of tried to trick them with some of the conceptual and context problems. Because we had just done replication, transcription, translation. So I threw in an RNA target, and half of the class was okay with a transcription factor having an RNA target instead of a DNA target, even though they're seeing DNAs. So these kinds of questions also, I think, help to figure out where they get confused. But then I was able to bring a sort of justification type of idea that I normally, again, try to do with free response by just having them annotate these figures. So just to sum up what I was going with AI versus, exam design, I used AI to pull relevant information and also match it with the kind of focus I had in class for each unit. I did all the figures, but I also asked it very specifically first to give me a good response that I okayed, and then I told it, put 10 mistakes in it, 3 of this type, 5 of that type, that kind of stuff, which didn't completely take the burden off of me, but I was more pleased with the outcome than when I sort of struggled 2 days before the exam. So I think I'll stop here and see if you guys have any questions.
Thank you, Didem. This is very interesting. Questions from the group? Emily, go ahead.
Hi, thanks. So can you say one more time how it does when you like give ChatGPT or Gemini or whatever the problem and how does it do if you feed your question to it?
So last year, and I can tell you the difference literally from one spring to the other spring, the first glucose transporter, when I had it and I gave it as a whole, first of all, it just bypassed the molecular structure. It just got the keywords and gave me an answer that was very vague. This semester, actually, and I also went in and bought the paid version to see what it can do. It was able to tell me the side chains and find ubiquitin side chains, like literally tell me this was lysine and it's not arginine, it's lysine. So its answer is extremely correct from the correct perspective. And I think that was a problem because now I mean, problem if I just gave it out to the students to practice. But it helped me because actually, Emily, this is a good question. I asked it things and I said, say it in 5 sentences. I want this and that in each sentence. If I don't direct it to be molecular and chemistry oriented, it tends to be much more biology. It loves, like our students, it loves the words, right? So I get like 4 sentences about ubiquitin, what it can do, beautiful things in the cell, even though I said, give me protein structure in it. So I had to, I guess I was spending almost as much time as I would spend with a student to get it to go into that structure. But once you have it, it's very good. It can also make tables for you, count things from students' mistakes, these kinds of things. So if you can feed all those things and say, okay, now knowing this is where my students are having mistakes, tell me 5 different ways you can make mistakes. And it generates those options really, really well. That's the part I liked. So yeah, I think right now I don't have, the only place it still fails for me is interpreting graphs, especially if your y-axis is on logarithmic scale. It cannot figure that out, but I'm sure it will soon. So I think, I mean, I did some enzyme kinetic things. I guess one last thing I'll say here is, it is at a point right now, if any one of you try one of your favorite questions, you will get sort of one of your good, clever, but not fully, an answer that is like someone who didn't study as much as they could, but had a big potential. So throwing these things, being very confident, you know, making it look good, we are there. And so it gives a very good idea of where you can hide these kinds of things. And again, I'm trying to find a way to make it so that I could also check students' reasoning skills by doing this. Basically, I'm trying to tweak AI to make reasoning mistakes now, not correctness mistakes. And I have to say, I've been doing some side work with trying to train AI on other things also. So on that platform, I had some privilege of getting to see how they reason, like how it reasons out, and it taught me a lot. So I think that I'm trying to build in to see if we can get, I can get my students of 200 people in class to really force them to look for those, not take it face value what they're reading, or really question if the logic is correct. Thank you.
Other questions? Josh, you wanted to say something?
Yeah, Didem, thanks. This is really cool. You were mentioning how the AI could predict how, like, wrong answers. How well did those correlate to students' actual, misguided answers or something?
It is not bad, Josh. I mean, I think it really is, it depends, as we all know, what we tell it. So if you if I tell it, so if I give sort of some information about my class, what kind of students there are, and some other things that I do, it can hone in more to the most likely type of things that they're going to do wrong. I don't know where it's sourcing that information, but I can also help it by giving some of my old answers to it. So I can feed it.
And it will give you a word-by-word wrong answer, or is it saying these are generally the things the students would get wrong?
No, I usually ask. I say, what do you think this kind of student, where would they be giving wrong? Or knowing that I have this kind of students, or we covered this in class, put something that you think would be less likely be caught by a student or something like that. So I give it the directive to change it, or give me a list. So for example, it knows when I say, for example, Saltbridge question I was giving, it didn't pick any random amino acids. It tells me to change lysine to arginine, for example, and glutamic acid to aspartic. So it still looks like a correct salt bridge, but not the one that's shown on the figure. And a lot of students flew by it. Like everybody was thinking this was correct because it was a salt bridge they knew. It just wasn't the one that was on the figure. And that's one thing I'm trying to train them. You have to interpret the figure. If I'm putting the figure, it's there for a reason.
I like this approach of starting from the mistakes and working backward. It's kind of a new definition of backward design. So.
And I think I do have to say, I did not get a complaint for this type of question. I was shocked because they hate many new things I try in class because it gets them out of their comfort zone. And all I told them was, because I mean, I'm known in the school right now. I'm one of the very few people who are left, who still have written free responses in their exams. So I get a very big pushback. So I told them, hey, there is still going to be free response part to your exam, but I'm replacing this semester one of them with the answer that you need to fix. And they were like, cool. But I thought they would think that was the easiest question. They don't. They feel like, because it restricts them, they can't write everything that they have studied, right?
Yeah.
So from an instructor perspective, I could not recommend this more to anyone who's teaching big classes. It really assesses, I think, and it is a breeze to grade. I mean, you can really give undergrads this thing to grade, because I had a few very, very meticulous student who had a tallying sheet for every one who was grading. So she was going 1, 2, 3, 4 mistakes. Okay, and the way I would do is if you have 8 to 11, you would get 1 point. So it would be windows, so not 1 point per mistake, but really group them. If I usually put in 5 sentences, somewhere between 10 to 14 mistakes. So for that 10 to 14, if they got at least 10 of them, they would get the full proficiency. But I also deducted half of that if they incorrectly, made one incorrect by their correction. So
Okay.
I think it ended up being discriminating between people who really knew what they were doing versus people who were just changing everything so that they can hit, because then they can't hit that.
Yeah, that's cool. Thanks, Didem.
So Didem, I wanted to ask a question about the time commitment. So clearly, um, as your students have shown that, even though it appears to be easy, you really, really, really need to know what's going on and the structure and the function in order to pick out all the right answers or pick out all the wrong answers. How much time is it taking you, or has it taken you over these couple of years to get to a point? So if there was someone else in this group, or another teacher who wants to take on this approach to designing questions, do you think this is like, oh, we spend one afternoon and we'll be done, or this is going to take weeks and months of practice in order to get there?
Everybody is different, but I do have 2 tips if anybody wants to try it. One is, I think it's also very important for the students to get used to a certain style and buy into that. So don't do just 1 or 2. So, whatever you're going to do, you'll need to keep up. There were times I said, why did I change my structure? Because now I do have to create this for every exam. However, the other thing is, this worked, at least for me. What saved the most time was starting with an open-end question that I already built and I liked, and I was aware what kind of mistakes students made. So just for full disclosure, a lot of the questions that worked best were, some of you know, at first I was 10 years at Wellesley Small Liberal Arts College, then I moved to BU, another 10 years. The first 10 years, I went to those exams because those were the ones that I had, from 15 years ago. I knew my students well. They were not written for the current students. They were more open-ended, where we were expecting the students to figure things out. As years went by, I was trying to be, because they would say, I didn't understand, what were you really asking? So I had started becoming extremely prescriptive with the students. The older ones were more free response. So those worked really well. If you want to try it, that's what I would say. Go to something that you know what students do. Just use AI to reprocess it and change the structure, put it into a format. And the other thing, again, is for someone like myself who can't repeat the same recipe twice, AI is great because I committed to this structure and I asked AI to give me for each unit this type of structure, which I think the students really liked.